Welcome to this lecture on developmental biology. Today, we are going to explore one of the most miraculous and critical milestones in human reproduction, embryo implantation. We will understand how a tiny cluster of cells manages to anchor itself and signal its presence to the mother's body. To begin, let us imagine you are trying to plant a delicate seed in a garden. You cannot just throw it on hard concrete. It needs soft, nutrient-rich soil to take root. In human biology, the embryo is that tiny seed, and the thick lining of the mother's uterus is the soil. The embryo must gently land, stick firmly, and burrow deep inside to connect with the mother's blood supply. If this does not happen perfectly, the seed simply cannot grow. Now that we have an intuitive picture, let us look at the formal biological definition. Embryo implantation is a highly coordinated process where a mammalian blastocyst attaches to and invades the endometrial lining of the uterus. This remarkable event does not happen immediately. It typically occurs around 6 to 7 days after fertilization has taken place in the fallopian tube, giving the embryo time to travel and develop before it is ready to settle. This brings us to the specific steps of the process, which is divided into 3 sequential stages. The first stage is apposition. During this phase, the blastocyst loosely rolls along the uterine lining, almost like it is searching for the perfect, receptive spot to settle. Once it finds that spot, the second stage, adhesion, begins. Here, specialized molecules on the surface of the embryo lock together with receptors on the uterine wall, creating a firm and secure physical bond. After the embryo is firmly attached, it must secure its survival. This leads to the third and final stage, invasion. During invasion, the outer layer of the embryo, known scientifically as the syncytiotrophoblast, aggressively burrows into the maternal tissue. This might sound intense, but it is absolutely necessary. By invading the tissue, the embryo establishes a shared blood supply with the mother, which is critical for the exchange of nutrients and oxygen required for future growth. Before we move on, let us see this process visually. On the screen, you can see the 3 stages we just discussed, moving from left to right. First, on the left, apposition shows the blastocyst gently resting against the pink uterine wall. In the middle, adhesion shows a much tighter physical connection forming. Finally, on the right, invasion illustrates the outer cells of the embryo reaching deep into the endometrium, tapping directly into the red maternal blood vessels to secure a lifeline. Having seen how complex this process is, it is easy to understand why it is the ultimate bottleneck for a successful pregnancy. In reality, nearly half of all fertilized eggs fail to implant correctly, making it a leading cause of early miscarriages. This knowledge is the absolute cornerstone of modern reproductive medicine. In clinics performing in vitro fertilization, doctors must carefully monitor the thickness and receptivity of a woman's uterine lining to time the embryo transfer perfectly, maximizing the chances of it taking root. While timing is critical, location is equally important. Sometimes an embryo implants in the wrong place, such as getting stuck in the fallopian tube instead of reaching the uterus. This results in a condition called an ectopic pregnancy. Because the fallopian tube cannot expand like the uterus, this is a life-threatening emergency for the mother. Understanding the mechanics of implantation is therefore not just about fertility, but also a vital part of maternal emergency medicine. Once the embryo successfully implants safely in the uterus, it immediately begins its next task, communication. Its outer cells start secreting a crucial hormone called human chorionic gonadotropin, or HCG. This hormone acts as a biological signal to the mother's body that a pregnancy has begun, stopping the normal shedding of the uterine lining. In a healthy early pregnancy, the concentration of HCG in the mother's blood grows exponentially, doubling every few days. This rapid growth is what doctors look for. This exponential growth we just discussed can be modeled mathematically. The formula is h of t equals h0 times 2 raised to the power of t divided by d. Let us break this down. H of t is the total hormone concentration at a given time. H0 is the initial baseline concentration on the day of implantation. The variable t represents the elapsed time in days, and d is the doubling time. If the doubling time d increases, it means the hormone is rising too slowly, which can be a clinical warning sign. Now that we have our formula, let us see this with a concrete example. Imagine an IVF patient whose initial blood test shows an HCG level of 25 units. The doubling time is exactly 2 days, and we want to find her expected level after 6 days. First, we calculate the number of doubling cycles by dividing the 6 days by the 2-day doubling time, giving us 3 cycles. We plug this into our formula, meaning we multiply the initial 25 units by 2 raised to the power of 3, which is 8. Multiplying 25 by 8 gives us a final expected concentration of 200 units. This tells the doctor the embryo is growing perfectly. Let us look at another practical scenario, this time working backward. A home pregnancy test requires a threshold of 100 units to show positive. If a woman starts with 12.5 units and the doubling time is 2 days, how long must she wait? We set our target of 100, equal to 12.5 times 2 to the power of t over 2. Dividing both sides by 12.5 gives us 8. Since we know 2 cubed equals 8, we can match the bases and drop them, leaving us with 3 equals t divided by 2. Multiplying by 2 gives a final answer of 6 days. This is exactly why doctors advise waiting before taking a test. To wrap up, we have covered the full journey of embryo implantation. Remember that this miraculous milestone requires perfect timing and precise cellular communication to succeed. It unfolds in 3 distinct stages: apposition, adhesion, and invasion. Once attached, the embryo secretes HCG, a hormone whose exponential growth serves as the primary indicator of a healthy pregnancy. By mastering these concepts, you have bridged the gap between basic cellular biology and real-world reproductive medicine. Great job following along.